Mutations. Okay, so what is a mutation? Mutation is a change to the nucleotide sequence of DNA. So these happen spontaneously. They happen anyway. Think about handwriting a copy of an encyclopedia. You're going to probably, no matter how hard you try, make a mistake at some point along the way. So DNA replication is very accurate, but it does make mistakes. Most commonly, they're going to ask you when or which phase of cell cycle does this happen in? Well, that's going to be interphase. The reason, because this is when DNA replication takes place, it's also the longest phase of cell cycle. Another word you're going to need to know is mutagenic agent. Okay, a mutagenic agent is anything that increases the rate of mutation to above the normal levels. There's three main types that you need to know about. So we've got base analogs. Analog means like analogous, similar to. So it's a chemical that can substitute for a normal nucleotide base. So it sort of takes the place of a normal nucleotide. This is generally going to cause a substitution mutation. What else can be a mutagenic agent? Well, radiation can. Radiation, it can cause basically weird bonding to go on inside the DNA, so it can change the structure of DNA. And obviously, if the structure of DNA has been changed, when you come to copy it, DNA polymerase won't know exactly what to do. The examples of this, we've got radiation, such as UV and ionizing. And the other thing we can do is change the DNA bases. Okay, well, these are chemicals that are going to basically move inside the nucleus and react with DNA to change its structure. These are going to cause either substitutions or deletions. Possibly additions as well, but we're going to look at the types of mutations next. Okay, we'll start off with the most straightforward one. We've got a substitution. Well, quite clearly in the name, one base is swapped for another one. So I'm going to use a little strand of DNA here, and we're going to look at what changes it goes through when a substitution takes place. So if we've got GTT, I'm just making this up. I'm going to do them in sets of 3, CAG, and then CTG. Okay, so this is what I'm going to start with each time. And let's say a substitution takes place on this C. So we're going to GTT, unaffected, but instead of being a C, it's now, let's make it an A. It can pretty much swap for anything. And then the rest of it carries on as normal. So we have another A, G, C, T, G. So what can substitutions do? Well, it's possible that a substitution can have no change because DNA is degenerate. This means that there's more possible combinations of codons than there are the number of amino acids. So you might change from, what have we got? Change from CAG here to AAG here. Possibly that goes for the same amino acid, and therefore there'll be no change to the polypeptide. It could also cause a single amino acid change. A single amino acid change, this could be catastrophic. It might destroy the shape of the active site. Maybe it's no longer complementary to the substrate. It could be beneficial. It might make the active site more complementary to the substrate. Or it could be neutral and effectively have no change on the fitness of the enzyme and therefore the fitness of the organism. Next up, an addition. So pretty straightforward by the name. Sometimes it's called insertion. So it's the same thing. An extra base is added. Well, what's this going to look like? For an addition, I'm going to start with my same sequence. So here we've just added one in, pretty straightforward. I'm going to put deletion down next as well, because it's basically the opposite. So this is when a base is removed. So in this case, again, we're going to start the same sequence. There's obviously no nucleotide that has an X, so I don't mind putting a cross in there. You're not going to get confused with that by now. So that just, this first T is gone. So what can these guys do? Well, I'm going to put both of these together. What happens with an insertion, an addition, most examples call them insertions, AQA go with addition. They can cause a frame shift. And this basically means that all of the following amino acids or triplets in the sequence are going to be affected. So a frame shift takes place if you change the number of nucleotides by any number that's not divisible by 3. Obviously, a triplet is in itself 3. So if you add or take away 1, 2, 4, 5, 7, 8, any number that's not a multiple of 3, you're going to cause a frame shift. And that's generally going to have a catastrophic impact on the polypeptide, because the chances of you changing 1,000 amino acids in a row and them all being a beneficial change is effectively 0. What else have we got to look at? We can look at an inversion. So an inversion is when a sequence of bases are reversed. So in this example, again, I'm going to have my starting sequence. So it's going to change, and let's take from this T to this C. So we're going to start with G. I haven't included G in that, so we're going to start with the G. But now we're going to read this one backwards. So it goes CGA, CTT. And then where do we pick up afterwards? So we've all got another T and G. So what impact can this have on the amino acids that are coded for by them? Well, it's possible if it's a small inversion, because of the degenerate nature of DNA, we can have no change. That is a possibility. It can change one or a few amino acids. And think to yourself, can it cause a frame shift? You're swapping the order. You're changing the sequence, so it can have an effect on the amino acids, but it's not going to cause a frame shift because you're not changing the total number of nucleotides. Okay, 2 to go. So next up, we're not going to write too many more notes on these. We've got duplication. What do we need to know about duplication? Well, duplication sounds a little bit like addition, but it's not quite the same. So it's when one or more bases are repeated. So let's look at this. Let's do our starting sequence. Again, we're going to start with the nothing change, so GTT. Let's repeat this AG here. So we've got C, and then we've got maybe AG, AG, AG. I'm not going to do any more. It could be many more repeats. It's definitely possible. And then we resume afterwards. So C, T, G. If this here, I've done 3 repeats of 2 bases, which gives us 6. So this will not cause a frame shift. But if I'd done 2 repeats, then I would have had 4. It's not divisible by 3. It would cause a frame shift. So I'm not going to write any impact notes here because it totally depends on the change that takes place. And the last one you need to know about is translocation. Again, the names are going to give you a big clue to this. So this is when a sequence of DNA is removed from one part of the genome and moved to another part of the genome. And that could be on the same chromosome. It could also be on different chromosomes. Okay, so there you have it. That's the full range of mutations that you need to know about, what they are, how they work, and my best guess of what the question is going to be in terms of how they're going to ask it.